The overall goal of the following experiment is to demonstrate the use of image stream technology to quantify cellular mechanisms of primary immune cells from well-defined patient cohorts. This is achieved by first isolating peripheral blood mononuclear cells, or PBMCs, from blood samples acquired from patients or healthy volunteers. After isolation, the cells are activated and then fluorescently labeled. Next, the cells are hydrodynamically focused, excited with laser light, and imaged on a time delay integration CCD camera. Finally, images are acquired using the image stream cytometer, and quantification of NF-kappa B translocation in each cellular image is performed using the IDEAS software. Ultimately, the differences between each subject group can be determined through analysis of dot plots, histograms, and cell images.
The advantage of the image stream is that it combines FACS and immunofluorescence. We're able to look at cell signaling pathways in a lineage-specific manner, either single cell or population, and starting with only a small sample of blood.
This method can help answer key questions in immunology by localizing and quantifying signaling intermediates. For example, it can be used to investigate action-mediated nuclear translocation of transcription factors, or to look at the effects of aging on cell biology.
Lida Duan from my lab will demonstrate the technique.
After isolating peripheral blood mononuclear cells obtained from human volunteers, resuspend the recovered PBMCs at 3 × 10⁶ cells per 0.6 milliliters of medium